Can you hear me? Okay. Welcome. Happy New Year, everyone. Thanks for coming out before the snow to the first PBR of the semester. So it's my pleasure to introduce Dr. Michael Murrell, who is an associate professor in biomedical engineering and physics at Yale University, where he directs the Laboratory of Living Matter, which I've always liked the name of that lab. He received his bachelor's at Johns Hopkins and then a Ph.D. at MIT, where he was a presidential fellow, and did a postdoc at the University of Chicago, as well as the Institut Curie in Paris. Last year, he was inducted as a fellow at the American Institute of Medical and Biological Engineering. He's published in a number of great journals, Nature Comms, Nature Physics, Comms Biology, Cell Reports, on his work. And he was awarded the Arthur Greer Memorial Prize from Yale University for his outstanding scholarship. I'm excited that he's here because, so my background is in chemical engineering, and I was always more of a transport and flow person, which I'm excited he's talking about that today. But I saw him give a talk applying thermodynamics in biology, and it made me finally get thermodynamics and appreciate it, whereas before I never had. But he said he's not going to talk about thermo today, so it's okay, but he is going to talk about flow, which is one of my favorite topics. So I hope you all enjoy, and I'm really happy that you're here today.
So the focus of my group is on trying to understand how physics plays a role in many essential cellular behaviors. We're interested in cell division, cell migration, cell shape change, but we come at the system from a slightly different perspective. We focus a lot on the mechanics of cells and tissues, the role of interfacial tensions, bulk tensions, thermodynamics, other things that can potentially play huge roles and underappreciated roles in major essential biological phenomena. And so what I will try to convince you of today is that while we all can appreciate the role and the importance of cell migration, things that are important for early development and cancer, that actually there's another source of motion that we call convective motion, which can arise from large-scale gradients in pressures and surface tensions that occur on scales larger than what we see by individual cells. So again, most of what we understand about migration is that one of the most common modes of migration is mesenchymal migration. Cells can reach and grab and pull on their extracellular matrix like you see here. This kind of mesenchymal mode has been thought to be responsible for a lot of invasive activity. But there are other kinds of motions. We know that in early development, we can see a lot of different types of collective motions, like you see here in the C. elegans embryo or here in Drosophila. Now, some of the movements that we see are characteristic of what we might expect happens by collective cell migration, cells reaching and grabbing and pulling, but doing that together in a group, but others aren't. So if we actually look at the rate at which some motions happen, for example, with mesenchymal kind of motion, we get things on the order of a cell length or two, say, per hour. And while that might characterize on average what happens in developmental cases, we know that some motions can be very rapid. For example, if you look at this kind of initial swirl that you have here, it looks quite fluid-like and it can be very rapid. And so we ask, are all motions that we see originating from cell migration, or can there be another source of motion? And so one of the areas that we look is to understand, can we build forces on larger scales than what we see at the individual cell scale? And can the result of those forces be rapid flows? And so I'll begin very basically by describing what we know about how individual cells generate forces. You might be very familiar, but there's a method called traction force microscopy. With traction force microscopy, a cell is going to adhere to a polyacrylamide gel. This gel is very elastic. Inside of the gel, there's these small particles, fluorescent particles, so that when the cell contracts or moves, you can estimate the forces because we made the gel, so we know how rigid it is, and we can image the displacements. And so knowing how rigid it is, knowing what the displacements are, can allow us to back calculate the forces. Now, these are traction forces, also known as surface forces, but there can be other forces at play besides just these tractions. So in addition to these tractions, we know that a cell can also pull up on its environment. And so here in this great work, what they saw was that a cell would kind of embed into a really soft extracellular matrix mimic, like a polyacrylamide gel that was, say, softer than the cell was itself. It'll kind of embed, and then it'll pull up. It can push down in the center, and it can pull up on the sides. And so these are normal forces, things that can occur perpendicular to the surface, that are going to influence the balance of forces that are going to influence migration. So we can describe that a bit, because indeed, while cells can exhibit these behaviors, actually so can passive objects. So here you see a bubble. That bubble has a pressure, a Laplace pressure, a pressure across the surface, and it also has a surface tension gamma. And so these can be related by the law of Laplace. The pressure is going to be equal to 2 times the surface tension divided by the radius of this sphere. And so the surface tension for a bubble is a constant property. It's what we call intensive. And the pressure is going to depend on that. So if you've ever blown, for example, soap bubbles or something like this, you might remember that the big ones were kind of really floppy, but the small ones are kind of really round. That's because the pressure changes with the size of the system, not the surface tension. That is constant. But now we can think about cells kind of like bubbles, except not ones that are passive, but ones that can consume energy, can consume ATP, and can do things like assemble the actin cytoskeleton, generate stresses by myosin molecular motors, move ions across the membrane, thereby changing pressure gradients. And so it can use some of its contractile osmotic pressure differences in order to generate forces in that normal direction. So even if you had just a liquid droplet, like a water droplet on a surface, a water droplet on the surface will reach a contact angle that's going to be a balance of interfacial tensions between the droplet and the surface, between the surface and the air, and the droplet and the air. But if that substrate was really soft, then now that surface tension is strong enough to pull upwards. Likewise, the pressure that can come from that surface tension is strong enough to push downwards. Now, the relationship between surface tension and these other quantities is known as the elastocapillary length. So I mentioned that this was only for a soft substrate, what we call a gel. And so the magnitude of that stiffness is given by the elastic modulus E. And so that tells us the length scale over which these normal forces matter. So again, using the cell cytoskeleton, using ion channels and osmotic pressures, you can generate these forces in the upward direction, and the extent to which that system is deformed is a balance between how strong you pull up with the surface tension and how stiff the substrate is that you push down. And so this is what we call elastocapillary effects. Now, might this be relevant for biological scenarios? We know, for example, that the ECM can be soft, can be on the orders of hundreds of pascals. And so actually, if we compare that to the known surface tension of cells, we can actually get elastocapillary lengths on the size of cells. What that means is cells are strong enough to generate surface tensions that can deform their extracellular matrix, not only in traction, but also with regards to these normal forces. And so maybe it plays a role in invasion, in early development. So how can we understand the role of these additional forces? Now, so far I've pointed out 2 issues. One is that there's this speed paradox. I said that cell migration can be slow. When we look at a lot of developmental cases, actually on average, the migration speed or the speed of motion is comparable to single cells. But every now and then you get these really rapid motions. What can account for that? And then two, I just said that cells are strong enough to generate these surface tensions that can deform cells, can deform the extracellular matrix in the third dimension. So can these actually have the same end goal? Can these explain each other? So what we want to understand is, can gradients in surface tensions and pressures, things that we don't often think about for traction against a surface where a cell migrates, can they drive rapid motions? How does the motion that you get from that, what we'll call convective motion, how does that coordinate with actual migration? Because we're saying both of those things coexist. And can convection actually play a role in metastasis? So I'll use a variety of model systems, but they'll all be quite similar. We use cell aggregates, cell spheroids as model systems. Now, we are going to use a couple of different kinds. First, we're going to use sarcoma cells. Now, these have been engineered to have E-cadherin-based contacts. They don't usually express very high E-cadherin contacts. They've also been engineered to not produce ECM. And we do that from our physics perspective because we don't want the system changing over time, producing more of its own extracellular matrix, so on and so forth. But then also we're going to use a glioblastoma model in order to understand the role of these large-scale surface tensions and pressures in driving metastasis into extracellular matrix mimics. So what you can already tell by the fact that this is a spheroid is that it's round, and that's implicative of surface tensions because surface tensions will want to make things round. So we ask, if the forces already are strong enough to make this into a sphere, can the forces that generate this surface tension when we have a gradient in surface tension thereby drive motion? Okay, so what are these quantities that I'm talking about? So we can measure the surface tension of these spheroids. Here you see a sarcoma cell spheroid. We're using a technique called micropipette where we apply a pressure to it. It gets sucked in. It's going to come in over time by some length L. So it comes in initially very quickly and then more slowly. And then as the pressure is released, it backs out again initially quickly and then more slowly. Now, you might be familiar, the kind of rapid times that you see is more indicative of elastic-like behavior. And the slower dynamics are usually more indicative of a viscous-like behavior. So this is a viscoelastic spheroid, which will exhibit either elastic-like behaviors at short times or viscous-like behaviors at long times. Now, you can take a look at the rate coming in and coming out, and you can estimate certain parameters like the bulk elasticity, the viscosity, but also the surface tension. And so here, we're doing an experiment that takes about 2 hours. We can compare that to the viscoelastic time, how quickly you go from elastic-like to viscous-like, and that's on the order of just a few minutes. And so the experiment timescales are a lot longer than the time it takes to switch from elastic to viscous-like, and so we're going to treat it like a liquid droplet. So if we measure the surface tension now using this method, we find that you have something on the order of millinewton per meter. So water should be something on the order of about 100 millinewton per meter. So that's a pretty high surface tension. What's interesting, though, is that I mentioned for a bubble that the surface tension is constant. It's an intensive property. It shouldn't depend on how large the bubble is. Here, we see that small spheroids with a small initial radius have a higher surface tension than larger ones. So as the system gets larger, the surface tension decreases. Now, if we were to inhibit myosin motor activity using blebbistatin, we would find that the surface tension decreases. It's down to the millinewton per meter level still, but it's now constant. So when we get rid of that extra activity, now you get what you expect from a more passive system. Yep, I missed it. Yep.
Remain solid, or do they actually, does the central part of them either due to necrosis or to actually forming
Good question. So in the size scale that we're looking at here, they never get a necrotic core. That's actually pretty small. So they remain solid-like, but it still depends on how fast we deform them. And as you'll see, actually coming up, when we perturb them really quickly, we will still get fluid-like looking behavior, but the origin will be elasticity. So it all depends on how we probe them. In this case, we do an experiment, an hour, we suck it in, an hour, we let it go out. And so over these very long times, we can treat it like a liquid. But as you're about to see, there's other times when we have to treat it like a solid. So we're saying that surface tension seems to be something important here. It's round on the outside, it's round on the inside. Remember, surface tension wants to make things round. We see how large it is, but oddly enough, it depends on size. Now, can large surface tensions drive motion? So here what you see is a Petri dish with water, and on top of the water is pepper. And a little syringe with detergent is brought near it, and 1 drop of detergent is added. So this reminds me of growing up. I remember thinking of like the dishwater detergent movie commercials where you would add dish detergent to the surface of your dishwater and everything would move to the side. This is what we call Marangoni flows. Detergent will lower the surface tension of water. And so what that means is the surface tension here is immediately much lower than on the sides, meaning you're going to pull backwards. And so we just established that there was this large surface tension for spheroids. So if we created a gradient in that surface tension, can it drive motion? So here is a spheroid again. This is just the actin. So you can see this kind of like radial organization. But here you see a spheroid floating in solution. And where this asterisk is where we use a high power laser to kill off one or a small number of cells. So you have a cell spheroid. We're going to kill some cells, which means we're going to create an imbalance in the force. Before, everybody was pulling on everybody else. Now we've removed someone, meaning that someone is no longer there to keep that tension present. So what we see, either from this movie or one in which we overlay these velocity vectors, is that when we ablate, you have a rapid retraction because, again, now you have made this imbalance. Cells have to reestablish this balance. So you pull back on the sides, but you also move towards the center. So we'll let this play a few times, but you can see here at first, on the sides of the spheroid, you move back and the center moves forward. Then everything moves back on the sides and the center moves backwards. So you see how that now the center moves backwards, retraction, moving forwards, healing, going backwards. So if we were to freeze frame that, it would look like this. And so now what we have is this ablated system that exhibits multiple dynamical features. One is that upon ablation, as you might expect, you get this retraction. Something was generating this tensile force, you've removed it, but you actually get this motion that's quite consistent across the entire tissue, across the entire spheroid. A few seconds later, however, it all reverses. And so if one were to measure the velocity, we would first see it increase and then it would decrease. And if we integrated that velocity, we would see that you end up basically right where you began. But what's notable about this is not only do we have collective motions here, these are vortices, something that you would think of as arising more from fluids. But there's another thing that I didn't mention, which is that while we see these toroidal motions that are indicative of fluids, this is happening extremely quickly, on the order of tens of seconds. Now, I said that cell migration typically takes hours, but in this case, you can get collective motions across the entire tissue in only seconds. And in fact, there's 3 time scales. There's this initial retraction, tau 1. There's kind of a fast return and kind of a slower return to get you back to your original configuration. If we again inhibit myosin activity, the spheroid is still round, so it's got some surface tensions, but surface tension that's going to have to come from E-cadherin because the active contributions are gone. And when we do that, there's no flows at all. So again, if we look back at that original experiment where we decided to treat the system like a fluid, we said that there was this transition between solid and liquid, which was about, say, 5 minutes. And because the experiment took much, much longer, we said that it behaved like a fluid. Now, however, if we look at those time scales, tau 1, tau 2, and tau 3, all of those are significantly shorter than the viscoelastic time of 5 minutes, which means that all of these motions are not flows, they're deformations of a solid. And so you can have a solid system that looks like a toroidal motion in a fluid. It's an elastic version of that Marangoni flow. So we can actually, again, estimate material properties from this movie. So we can estimate what the surface tension gradient is. We have that vector field. We can project the vectors on the surface. We can take the elasticity that we calculated by micropipette, and we can estimate the now delta gamma. And just like before with micropipette, now we see that the surface tension gradient, again, depends on size. Small spheroids have a higher delta gamma, which decreases as the spheroids get larger. Likewise, that initial retraction speed, the velocity of moving backwards, is the highest for the smaller spheroids. They are inherently different than larger ones, which again is very different from how we think about passive systems. And if you look at that velocity, though, now we're talking about not only not microns per hour, but now we're talking about microns per second. And so we're able to move much faster than how we know single cell migration to work, a few different in vivo scenarios. We can now approach the rapidity that we see with other developmental events. So from this first part, we see that these simple tissues can have large surface tensions and pressures. Interestingly, they're size dependent. And when we disrupt them, we can generate gradients, and that those gradients can generate very rapid and highly collective motions. So this part was to establish that there are these large forces that we can have across the spheroids, across our multicellular systems. Can we show how this can play a role with migration and potentially with invasion? So I'm going to draw another analogy to fluids. Here is a movie in which a spheroid is placed onto an adhesive surface. So this is glass covered by fibronectin, and now a spheroid has been added to it. And so the cells migrate out of the spheroid in a way that might look very reminiscent to you of a liquid droplet spreading onto a surface. In fact, when a liquid droplet does this, there's something called a precursor film that comes out and wets the surface as the droplet is evacuated of its fluid. And really the same kind of thing happens here with cell aggregates. And so I introduced elastocapillary effects by saying, we know that the forces that cells can generate and the internal pressures that they can build can be really high. And they can be high with regards to the stiffness of the ECM that they're under. So we decided to do this experiment again, except one in which we vary the stiffness of that polyacrylamide gel and ask the question, well, if these forces are so significant, how does it influence motion? How does it influence the efflux of cells out of those spheroids, as a dramatically oversimplified way of trying to understand invasion? It's the physics version. So now we spread these spheroids onto a surface. We measure the area over time, A, divided by the initial area of a spheroid. But now we do this on polyacrylamide gels that can be made stiff, like 40 kilopascals, or soft, like 0.7 kilopascals. Now, initially, we see that there's really not much of a difference. And in fact, if we take an instantaneous rate they're about the same. Here's a spreading rate. It's something like a strain rate. We can see that the instantaneous value is not different between 0.7 and 40 kilopascal. Thankfully, however, my postdoc did so many replicates that we were able to notice a difference in its functional form. That on the rigid case, we have what looks like more of a line, that's the blue. But on the soft case, it looks, I don't know, perhaps more exponential. So we decided to take the difference in a fit between exponential and linear for spreadings on substrates that varied a lot in stiffness. And so what we saw was that you fit better to something exponential when the substrate is very soft, and you fit better to something linear when it's rigid. Now, could there possibly be an important reason why there's a functional difference when there's really not a giant quantitative difference? So we decided to explore this further by looking at forces. And so here's a movie of those traction force microscopies that I've been showing you, hinting at. On the left is a bright field image, so just white light. We also image the actin that's inside of the cells. And then on the right-hand side is our traction force movie. So there should be a bar that goes from 0 to about 100 Pascal. And so as the cells come out, we can measure the forces that they generate, again, by that traction, that kind of parallel force that was being generated. So this is for the rigid substrate. What you can see, and maybe I'll freeze it as it comes back around, what you can initially see is that, oops, nope, I don't want to give it away too soon. I like to build suspense. Nope, ay Dios mío. Okay, can't stop it. So the red dotted line is roughly where the sphere is, but again, there's a monolayer coming out. And what you can see is that underneath the sphere, there's not a whole lot of force. Peripheral, where the monolayer is coming out, they're tractioning, okay? So they're generating a lot of force, nothing confusing there. But if we did something softer, say 2.8 kilopascal, the sphere spreads. It actually spreads at almost the same rate. But now, if you look, the forces are a little bit attenuated on the outside, but they're higher on the inside. It looks a little bit more uniform. And so we can make a chymograph out of this data. And so we can show that the general pattern is that for rigid substrates, the forces are where that monolayer comes out, and they're very low underneath the spheroid. As it gets softer, the forces are kind of uniform. Now we're looking at the magnitude. We'll have to look at the direction in a moment. But as you get really soft, the forces are actually highest underneath the spheroid. Now again, same rate of spreading, but the manifestation of the forces are different. Here we have a stress gradient. And this shows that for soft substrates, the stresses are mostly under the aggregate, while for rigid, they're mostly outside of the aggregate. Again, same rate of spreading, but very, very different pattern of force. Now, exactly where you switch from the forces on the inside to the forces on the outside is exactly where we had that switch in the dynamics of motion. Okay, the seemingly irrelevant change from linear to exponential corresponds to a dramatic change in the forces. Now, I'm showing you the magnitude, but if we look at the direction, it'll be even more surprising. So for rigid substrates, all of the forces for the traction are inwards. They move out, they pull in. They move out, they pull in. But as the system gets softer, then you actually start seeing forces pushing outwards. So this is the kind of model that we have for that mesenchymal kind of motion. You reach, you pull, you grab. But in this case now, we have something expanding. We have something pushing outwards. So what can be the origin of that? So again, if we go back to these things called elastocapillary effects, we know that a droplet, a bubble, or also a cell spheroid can pull up and push down. So here you see a confocal slice. So you're looking at the spheroid from the side as it's spreading. You can see that cells are starting to come out here, but you might also notice that it looks like these cells can dip a little bit beneath the surface. And so what they're doing is that if we look at the level of the substrate over time, right as soon as the cell attaches, it's at 0, it's flat. But over time, the center of the gel gets pushed down and the side gets pulled up, just like what we would expect for a water droplet. So we're going to have a couple new parameters here. We have an indentation depth for Z, and we also have a meniscus height. So we can actually measure these quantities and calculate what the surface tension is. So with a cell spheroid that is on a soft substrate, again, it can pull up with a surface tension gamma that we've seen. We know that it pulls inwards because that's the traction force pulling inwards. And so we have a force balance here. And if we were to calculate what that gamma is by the force balance, we would see again that it depends on size. But actually it doesn't depend on stiffness. That's the blue gamma. So you might see where I'm going with this. We are interested in understanding what role these elastocapillary effects can play in motion. And so this picture paints a perspective that there's something else besides migration. Why is that? Because I mentioned from that law of Laplace that the pressure depended upon the surface tension and a curvature, this R naught, the radius of the spheroid. But now actually there's two curvatures involved. There's the one for R naught, which is the spheroid, but there's also one, for the radius of indentation. So if you indent and pull up, there's a second curvature here. So what that means then is that you've got two pressures, one that's acting on the bottom and one that's acting on the top. And so you can calculate what those pressures are, and we can see that there's a difference in pressure top to bottom only when the substrates are very soft. As the substrates get stiffer, because they can't pull up anymore, you can't generate that curvature. If you can't generate that curvature, there's going to be no difference in pressure. So the question then is, if on soft substrates, we have a difference in pressure, can that difference in pressure drive a flow? So what we decided to do then was to make a model, a force balance model, where all of these values are inputs now. We input our velocity field that was basically the same. We input our force field, and we try and back out what the pressure was that it took to get that type of motion. And remember, there's one signature, especially of pressure, that we're looking for, which was we always saw that pressure was size dependent. So do we see that? So if we now estimate this pressure, we can do it as a function of radial distance from the center of the spheroid. The pressure is highest at the boundary where the outside of the spheroid is. And on rigid substrates, if we input the forces, we input the dynamics, but in this force balance, we back calculate the pressure, we find a pressure, but one that does not depend on size. Whereas for soft substrates, when we back out that pressure, now we find one that depends on size, suggesting that that pressure was really important for that motion. Yeah. Exactly. Yes, it is. Yes, exactly. Yes, it is. All within, factor 2 or 3. Yeah. And so to summarize, we began by seeing what looked like a really kind of irrelevant quantitative difference in motion in the rate of cells coming out, what we're calling spreading or wetting or whatever you want to call it. But now we see that there are capillary effects. Here, you deform in 3 dimensions your substrate. Your surface tension of your spheroid is strong enough to pull up and to push down. Why might that be important? Well, because pushing down using pressure has a size dependence. For more rigid substrates, we say, well, you don't really. You can't generate that curvature. You're still generating forces in that direction, but they're not generating curvatures, which means there can't be a difference in pressure. So if we looked at the spreading rate then for a rigid substrate, so this is just like a strain rate, so like how fast you're coming out, we see that now if we take that data that we had just grouped together, but now we plot it versus the initial size of the aggregate, there's no difference. That's when capillary effects don't matter. But now if we take this data and we plot it versus the initial size, now we see a size dependence, suggesting that pressure matters here and doesn't matter here. And so this kind we're all very familiar with: cell migration, generation of traction stresses, and traditional cell migration. But in this part, we have to now think about convection because we know it's in large part driven by pressure. And so from that part, we show that gradients in pressure can drive motion on soft substrates. I'm skipping over a lot of other details, but other things that happen when substrates are very soft: focal adhesions are very small, they're very nascent, they're very transient, which also enables you to be pushed because you're not so stuck to the surface. However, on rigid environments, we have cell migration like we've always known it to be. Generation of traction stresses parallel to the surface. Oh, yes, please.
So I can't help but think that I'm a neuroscientist and I'm thinking about growth and development of the nervous system, or chunks of the central nervous system.
Yeah.
And areas are growing and expanding, and individual cells within there are expanding and pruning, etc. And of course, in the case of the nervous system, central neurons have elaborated, in many cases, processes that go beyond the spherical part that extend over incredible distances. And so as I think about the area that I'm familiar with in terms of growth and development of the area of the brain, we often see these kind of mappings that a part of the tissue is growing, but then the individual cells and their coverage of territory within there, which has very important functional consequences of the nervous system for sampling synapses and so forth, are growing at different rates. So they're covering relatively more or less territory.
Yes.
And I can't help but think that these two differences you just pointed out could be at play at those two different levels of the whole structure versus the individual cells.
I think so. And in fact, the last part, which is really just more of a vignette, is where we show that the formation of these projections and everything else can actually be kind of induced by these types of behaviors. To fast forward, I'll explain it more in detail, but essentially we show this in 3D, in which the surface tension in 2D involves pulling up like this. What would that look like in 3D? Well, in 3D, it involves pulling like this. And so one thing that we show is that these elastocapillary effects, even without any migration or the formation of any projections, can contract and align the ECM. And when you do that, then you begin to get these projections on the cell level. So it's actually not coming from cell migration that we typically think of, or I guess maybe it can also come from pressure and these other things, but that you can actually have tissue scale effects on the ECM that are priming individual cells for exhibiting these types of projections and other types of behaviors. Yes, please.
So there's an old experiment where, for neural crest migration, they took beads, so polyacrylamide beads, and replaced some of the neural crests, and the beads moved just like the neural crest. And so it sounds like that this may be part of the reason for that, because they had two differential rigid surfaces, the neural tube and the surface ectodermis.
Yes.
And then this massive beads, which have some adhesion attraction, but they're basically using the surface tensions to, because what was surprising then was that they actually moved like crest cells would move.
Yes.
And it was assumed that the crest cells actually just used their cytoskeleton and ability to recognize cues to actually, cognitively move.
Yes.
But this was a passive movement.
Yes, exactly. So actually, and that's the best way it's described as passive movement. It would be difficult in that kind of assay to exclusively show it's convective, because you can imagine cells all migrating, you're stuck to them, you're going to move with them. And so the question is, can you have motion in the absence of anyone migrating? Is it possible that the different balance of forces that you have can drive more rapid motion? So basically the framework we're coming at a lot of this is that we always think about tissues moving in terms of cell-cell forces and cell ECM forces. But to spread out, to migrate, we typically have to say that the stresses generated between cells and the ECM has to be greater. Because if it wasn't, they would pull off and all aggregate together. But that's exactly what we think is the type of motion that you have. Yep.
Mesoderm ingression.
Yes.
As well.
Yes, exactly. And just like that fast swirl I showed, you can get that if you have these larger forces, because cells can accumulate these forces, they can get large, and then as soon as there's a gradient, it has to relax. So part of the goal of showing the surface tension and rigidity of the surface is that by force balance, something has to happen. Like if you show that these two things are true, then you have to have this other motion. You have to find those cases where those are true. Yeah. So I come from, I guess I shouldn't call it so much fluid dynamics, not in the Munson way, but more in the wetting way, like liquid droplets. And so I was using this example of liquid droplets spreading onto a surface, but what would happen if the surface is confined and you're not able to fully wet, but you can only partially wet? What happens? And so here you see a spheroid on a fibronectin pattern. We chose donuts because it worked better, but we can ignore the fact that there's a center there. And so these spheroids will spread out to the surface, spread out to the boundary. But then what happens? We specifically wanted to ask the question, can we force convection to coexist with migration? And so what you see here is, again, the polyacrylamide gel, an image of a spheroid and the actin within it. So if you were to take a spheroid and drop it onto the surface, it's not able to fully spread out. Instead, it remains what we call partially wet. And what you might see, if not by the DIC movie, is by the velocity vector fields, that you have this persistent inward motion. So with bright field, with white light, what we're doing is we're integrating everything. You get everything that goes through the sample, a full three-dimensional picture. And when we integrate everything through the volume, it looks like you have this inward flow. If we were to freeze frame those, cells will reach out, like you see the red arrow. They'll try to extend beyond the pattern. They cannot, so it retracts. And then once it retracts, it seems to float backwards. So you can see how the contrast on the cell that's been highlighted changes. The reason that's changed is because it's no longer in the same focal plane. So while I think I'm leading you perhaps a little bit more than I should, you can see how there's a cell that is in focus that reaches outwards. It initially retracts, and you can see it's kind of out of plane, and it remains out of plane, moving backwards. And so what we show here is that cells are going to migrate to the outer boundary, detach, and be convected towards the inner surface. And so if we quantify this, and so what we're doing is we're taking two signals. One is the white light, the DIC, which is going to give us a signal across the entire spheroid. But you saw we also had those actin images. And so the actin is biconfocal, and so that's only giving us cells at the surface. So we have two things, cells at the surface and cells through the bulk. And we can, through particle image velocimetry, quantify the motion of both. And what we see is actually that at the surface, cells move outwards. They go to the boundaries. But then in the bulk, we see an inward flow. And so if we look at confined motion, we see that there's an inward flow for the bulk, an outward flow for the actin. If you were just looking at the wetting of spheroids without the confinement, they would both be outwards. But as soon as they're confined, you're not static, but instead you keep moving. You're basically cycling, or what we call idling. And so you migrate on the two-dimensional surface, but as soon as you hit the boundary, you are convected back towards the center.
Surface tension?
Yes, it strongly depends on the surface tension. Yeah, we haven't modified it. We haven't programmed in different levels of E-cadherin or anything, but absolutely, the convection is coming. So actually, thank you. The convection is coming from the surface tension of that curvature. I wouldn't have mentioned that if you didn't bring it up.
E-cadherin contacts are enough to allow
Yes.
for aggregation, but not so much to give a rigidity or increase the surface tension beyond a point where you couldn't get.
So at the surface, you'll have E-cadherin, which is absolutely essential for transmitting, but you also have the actomyosin stresses as well. So basically the idea is like once you're adhered, that tension is high. Cell ECM contact is very strong. And so cells are going to, wander about, eventually hit the surface. They're going to migrate outwards. But then as they try to reach outwards, they're going to lose some of that. They're going to destabilize because they've tried to reach. They cannot because it's non-adhesive out there. And now the surface tension gradients are strong enough to pull you backwards. And so you end up in this cycle. Yes, please.
I think that's more of a mechanical signal or something chemical, like, oh, this isn't an environment that I can.
I don't believe in chemical signals. Not kidding. But it's. So I would like to say that I consider it mechanical, principally, because the way I've pictured it here is that at this stage, because you cannot stabilize an adhesion, you now have only weak interactions with the surface. The ones that remain in the spheroid are still strong. And so your inability to stabilize something in a non-adhesive area will limit the cell ECM tension, while the cell-cell tension remains high and therefore pulls back.
Tension is still through integrin binding.
Yes, that's right. That's right. It's fibronectin on the bottom. Yep, it's fibronectin. Yes, please.
Direction. Do you
Yes.
get up in the middle? And is that because something about on the surface it's one, but then on the surface tension it's the other?
Yeah, great question. I'm not really sure why.
That's really cool that it sets up the same way.
I think I can speculate,
Sure.
which is my favorite thing, is that I think that the reason, initially you could be any one of these little Voronoi moving about. You're going to hit the surface. There is a bit of a density gradient on the side. I don't know if you noticed, but because you retract, you never actually fully get to the outside boundary. So there's a bit of a density gradient, which makes me think that you're always going to be driven out to that side because there's a lower cell density. And as a result, because you have this fried egg shape here, as soon as you de-adhere here, you're always pulled inside. And by mass balance then, that means that you should basically always have that kind of cycle. Yes, please.
Sandwiched and aggregate between two parallel matrices.
If you sandwiched it between two parallel matrices, I do not know. I can speculate. So if you sandwiched it between two things, then you're going to have another curvature again, because it's going to be whatever curvature you have on the side of your hamburger. And so you're going to set up a different pressure gradient. So maybe that'll give you the different pattern of flow, actually. Yeah, great question.
From fibronectin to laminin to polyl-lysine to collagen.
Yes, we have. So polyl-lysine, nothing moves because it's just glued. Collagen, if I remember correctly, nothing moves. I think because it's too sticky. And we can increase the fibronectin to the point in which nothing moves. So I think it just has to be that you can't be that much more dramatically attached to the ECM. Otherwise, that surface tension gradient isn't enough to pull you off. Yeah, that sounds about right. Yeah.
So the fibronectin, I know that there's a lot of different shapes. There's like crossbows and X's and T's and
Yeah,
F's
that's right.
and whatever else. Have you tried, you said like the donut looks best. And I get
Yeah,
that.
for no good
So,
reason.
well, maybe.
Maybe.
Maybe not, right? Like,
Yeah.
is there ways to think about, like, you can't have a stable system?
Yeah,
but
that's a
it's
good point.
T, and
That's a
you're
good
gonna be
point.
able to model that, like,
Maybe it is.
computationally.
Yeah, maybe it is for that. Because I remember my postdoc just said when it was just a solid circle, it just wasn't super obvious.
Yeah.
So yeah, maybe. That's a good point. Maybe that is, I really didn't think too much about that before.
Kind of like the generation of wind, where you have to have uneven heating of the Earth's surface to cause the convection variance.
That's deep.
I'm just holding them, I don't know anything about this.
Yeah, quite possible. So yeah, maybe so. Yeah, maybe so. Maybe that is involved. I mean, we couldn't really get anything to work on any other pattern. So that might tend to make you think it's essential. Just from that short thing, if we can get convective and migration, convective motion and migration coexisting. Interesting, you don't get kind of a dead dynamical system, you get one that is what we call idling. And so for the very brief final part, we also did this in 3 dimensions to try and assess what the role was of large-scale surface tensions and pressures on migration into the extracellular matrix. And so actually what you're seeing here is a spheroid that is in a 3D polymer gel. What I want you to focus on, which I'll explain very briefly, is the fact that, again, you see that radial inwards motion. So this is a particle image velocimetry on a spheroid in a gel, but you also see an inward flow of the ECM. Well, or you have to take my word for it because it's not fluorescent, but you can see it under DIC. Prior to cells actually coming out, you see this motion. So we will work with glioblastoma as well. And in this case, we use a glioblastoma spheroid. We focus on the fact that the volume can change significantly with glioblastoma cells through regulation of ion channels. As I mentioned, size really matters. And so we wanted to manipulate the volume of cells to alter those properties like pressure and surface tension. And we know that these cells can alter their volume by up to about 30%, which is pretty big. And in particular, we're going to use an inhibitor of a ion channel cotransporter, NKCC1, sodium potassium 2 chlorides. So of course, this is going to change the concentration of ions across the cell, and water will follow suit. And so we can change the volume of cells by altering the activity of NKCC1. Now, as we all know, cells can align their extracellular matrix. This can lead to greater invasive capabilities, align and stiffen. Migrating cells are often associated with creating collagen fibrils. Much of this work was from one of my old colleagues at University of Wisconsin, Patty Keeley, who really kind of established the ways in which cells can communicate through the assembly of these fibrils inside these collagen matrices. And so upon the establishment of these strings of these long kind of bundled fibers, cells can migrate out faster. And this is what's been attributed to driving migration and invasion. We actually ask whether or not the cell migration is essential. So here we have a spheroid. In this particular case, the ECM is not labeled, but perhaps you can see a kind of a haze in the background. And what we've done here was inhibit NKCC1. The result of inhibiting that is that there is an influx of water into the cells. As a result of the influx of water, the spheroids also swell. Now, what seems to be contrary to the physics of the scenario is that the spheroid is getting larger. So you can see the blue arrows moving outwards, but the ECM is moving inwards. So ordinarily, we're used to the idea of a contractile spheroid contracts and pulls on the ECM. But how can one that's expanding contract the ECM? In cases where we do not have the inhibitor to NKCC1, the aggregate contracts and the gel contracts. But in the case in which we inhibit it, the aggregate expands, yet the gel still contracts. How is that possible? So this is a bit much. We used a computational model. We have an elastic mesh, one on which there's a cell, which is this region here, and one in which there's an ECM. And what we can do is we can change things like the elasticity of this inner region, the aggregate, versus the outside. We can alter the interfacial tension. We can alter the ability of water to move across the boundary. And we can alter what's called a contractile body force. Just for the sake of time, essentially, the only way in which one can get an expanding spheroid with a contracting gel is to have a strong surface tension at the inside, at the periphery, which is going to do the contracting. We allow water to move, the system contracts, the inside accumulates water, and the balance of that influx of water with the contraction of at the surface, or what we call these elastocapillary effects. Only in that case can we get the spheroid or blue expanding and the ECM or yellow coming in. If we only alter surface tension, it's all in one direction. If we only influence fluid flow, it's only in one direction. But when we do both of them together, we can actually get this effect. And so the idea was that we don't have to have cells migrating outwards to contract the ECM, that it can all be due to elastocapillary effects from the spheroid itself, and that they can play a role in priming the system. So I'll just end there. Overall, we really just want to show that what we call interfacial properties, surface tensions, pressures, can yield motion that can be faster and more correlated than migration. And that actually, in some cases, depending upon the magnitude of that surface tension and the stiffness of the surroundings, it has to happen. And so convection will need to coexist with migration. One can play a different role in different scenarios. So with that, I want to thank all of the awesome people I've gotten to work with. Most of these experiments are done by an extraordinarily talented experimentalist, Suleiman. A lot of the physics and the analysis by Vikrant, and a PhD student, Sarush. And I love working with a bunch of theorists. These are also my good friends, Nick at Cornell, Amir at Yale, so on and so forth. And I'd like to thank you for your attention.
So we probably have time for one or two questions. There were a lot of good questions.
So can you elaborate a little bit more about the role of these convective movements in metastasis, right?
Yes.
Because as the tumor grows, you get a lot of ECM. And of course, this may differ for different types of tumors. And so as the ACM gets stiffer, how much do you think these convective movements will contribute to invasion and eventually metastasis?
As the ECM gets stiffer, less, actually. So, and also it should be noted that the motion that we're really talking about is, while I said that, these things spread out at the same rate, whether it's migration-based or convection-based, well, really the motion that we're talking about is getting those cells to the outside of the spheroid, not really the long distance stuff. So it's basically in either case, soft or stiff, only able to get cells to the boundary of the spheroid. And then once they're out, they're out. So in terms of the metastasis, if the limiting factor is getting cells from inside the bulk to the surface, it's going to really matter. If that's not the limiting case, as would be the case the more rigid it gets, this mechanism would become less important.
Any other questions? So online, Arena Korneva, who's in BME, she said, what kind of technique do you use to track the I can't even say it.
Oh yeah,
toroidal
toroidal motion. So we use particle image velocimetry, a really common technique for fluid mechanics. So basically you compare two images and cross correlate them, and you can identify the direction and magnitude of motion.
Any other questions? I'll end with one last one then. I was just wondering so this flow in the spheroid, how that might correspond to flow within a cell
Yeah,
at the
good
same
question.
time. Like, do you think they're opposite? Do you have no idea?
Yeah,
Like-
actually, I've never gotten that question before. Because one might think that to exist in a force balance, yeah, maybe there is something opposite.
Because I know when like you get the grabbing motion, they have shown that like when the back releases, the flow all like pushes to the front of the cell.
Interesting.
But this is a different scenario where you don't have that if you're having convective.
Well, yeah, actually, that's a good question. I mean, if in the case where we have the inhibited spreading, I'd be willing to bet that there is a relationship between the internal flows for cells that are approaching the boundary versus the ones that are moving backwards. Because as you said, we kind of know what the ones approaching the boundary should be like. So actually, yeah, really good question.
Well, let's thank Mike for the great talk. And thanks everyone.